Hi guys, myself Akanksha. Welcome to another session of endocrine physiology. Today we are going to talk about a very important aspect of endocrine system, that is about thyroid gland and its hormones, how these thyroid hormones formed and where it gets synthesized in our body. The whole mechanism I will explain later on. So let's start with some basic physiology. First we talk about origin. So its origin is endodermal, and if we talk about location, then we know in the midline of the neck hyoid bone is present, and just below it we see the larynx, and just below the larynx the butterfly looking structure is seen, that is called thyroid gland, and it is bilobed. Bilobed means there are two lobes which is connected by isthmus, and just below it and just below the thyroid gland trachea is present. If we zoom into the thyroid gland, we see some special follicular cell surrounding a small area forming a colloid or a lumen. This space is called colloid or a lumen, and here blood supply is also present in between these cells. Actually, the thyroid gland is composed of many large number of closed follicles which are lined by cuboidal epithelium, and this follicular cavity or a lumen is filled with thyroglobulin, which is secreted by this follicular cell. And one more thing we have to remember, that this follicular cell is the only responsible for the secretion of T3 or T4, means thyroid hormone. T3 means triiodothyronine and T4 means thyroxine. So this follicular cell is responsible for the secretion of T3 or T4. Now let's move on into the synthesis part, how this thyroid hormone gets synthesized. We discuss this theory with the help of this diagram. Signal first comes from brain, then the hypothalamus in the brain, which releases the thyroid releasing hormone, which stimulate the anterior pituitary to secrete the thyroid stimulating hormones. Now you can see there is a TSH receptor on the membrane of the thyroid gland, and you can see if we zoom this area, you can clearly see this is your follicular cell and this is the blood capillary. This TSH receptor is attached to this G protein, and TSH gate binds to TSH receptor and transform the GTP into GDP, and as a result cyclic AMP level increases in the blood inside the thyroid gland, and this cyclic AMP gives signal to this sodium iodide symporter to transport the iodine into the follicular cell. Now this NIS symporter, which transport the iodide from the blood stream into the follicular cell along with sodium ion. This iodide is now inside the follicular cell, and there is one more transporter that is called pendrin, which transport the iodide from follicular cell into the lumen or the colloid, or this space is also called follicular cavity of the thyroid gland. Inside the follicular cell, endoplasmic reticulum form the thyroid globulin precursor, which goes to the Golgi body apparatus and form the thyroglobulin, which is composed of tyrosine chain. Now this iodide in presence of thyroperoxidase converted into iodine. This iodine get bind with this tyrosine chain and form the MIT and DIT. If one iodine is attached to the tyrosine chain, then it is called monoiodine tyrosine, and if two iodine is attached to this tyrosine chain, then it is called diiodotyrosine, and this process is called iodination. Now if one MIT and one DIT get attached with ester bond, then it is called T3, that is triiodothyronine, and if two DIT are attached with ester bond, then it is called thyroxine or tetraiodothyronine, that is T4, and this process is called coupling reaction. After the synthesis of T3 and T4, it get stored here, means in the lumen it get stored and attached with thyroglobulin chain, and then this is taken up by the cell by the process of endocytosis, and when it entered here, Then lysosome fuses with endocytic vesicles and releases the enzyme which hydrolyzed or cleaved the thyroglobulin to form the free T3, T4, MIT and DIT. Then this free T3 and T4 get released into the bloodstream again because they are not because they are lipid soluble, so they don't need any transporter, and they get combined with their binding protein, whereas the free MIT and free DIT get deiodinated and iodine get released, which further reutilized, and this is called deiodination. One more thing we have to remember here is that if the T3 T4 level get increased more than required in the bloodstream, then it will give negative feedback to hypothalamus. In this way, this whole mechanism get continued. Thank you so much guys, and I will provide the notes in the description box, and if you liked it, please guys do share with your friends.